So to continue with the types of muscles and their classification, okay? The very first topic will be mechanism and process of relaxation, contraction and excitation of muscles. मतलब कोई भी muscles, जैसे as we have discussed in the previous class, there are basically three types of muscles: skeletal muscle, cardiac muscle, and smooth muscles. So in today's class, we will review on the mechanism and various process of muscles like relaxation, contraction, and excitation. So how these muscles relax, contract, and get excited. So to begin with the first, that is a smooth muscle. The sense is that this smooth muscle have a special property that is known as plasticity. It is also referred as stress relaxation. So this smooth muscle, they have the capability to adjust to their stretched form without increasing their final tension. I mean to say, the muscle will retain or will come to their final position without having any pain or in the pressure exerted to the muscle bundle. So basically this smooth muscle is a hollow viscous which is surrounded by the various smooth muscle fibers, and the plasticity which allows the expansion of stretch. This is within the physiological limit, and definitely there should be no any pressure or pain will act on the muscle fiber to make it contract or relaxed. So this smooth muscle doesn't lose its contractility, and it's just believed that due to changes in the various arrangement of the myosin and actin molecules. Because as previous we have studied that for relaxation or contraction of any type of muscles, these myosin and actin filaments, these are very much responsible. So to keep the view regarding the phenomenon of this myosin and actin filament, the relaxation and contraction of each and every muscles will rely on these two filaments. So this is also believed that this connective tissue, they are surrounded with the specific muscle fiber, to a bundle of muscle fiber, and to a whole muscle, because it gives it extra capability for relaxation or contraction of the specific smooth muscle. So this connective tissue, they are also made up of this reticular tissue. I mean to say, they are having the effect of this collagenous plus the elastic fibers, and that will combinedly makes the connective tissue which is associated with the smooth muscle basically. So come to the next point. These smooth muscle cells, they respond to norepinephrine, which is released by the sympathetic nerves, and acetylcholine also, that is released from the parasympathetic nerves. So being they are antagonistic to each other, because they have specific work in a specific arrangement or in a specific situation. So whenever there is the innervation of sympathetic nerves, the norepinephrine will act and make sure to relaxation or contraction of the smooth muscles. While whenever there is innervation of parasympathetic nerves, they will tends to release this acetylcholine, and it will act on the smooth muscle bundle or fiber to make it contract or relaxed. So basically few smooth muscle there consists of distinctly separated fibers that require nerve stimulation to contract, that is called multi-unit smooth muscle, and it includes pilomotor smooth muscle cells, especially in the skin, iris and ciliary body of the eye. So if we talk about the self-excitation or the contraction of muscles that persist within the muscles, that is due to the ionic exchange of the sodium and calcium, because the sodium and calcium, they when they comes to the fiber region, they will cause the resting potential to decay down towards their threshold position. So basically the value is 35 millivolt, but while the resting potential is in between minus 55 to minus 50 millivolt, and the action potential, threshold action potential value is minus 35 millivolt. So definitely it will come in between the minus 55 to minus 50 millivolt. One thing is there also, that is the sodium and calcium, they are pumped back out before the threshold is reached. So the membrane resting potential is again restored. So to come the membrane potential in resting position, these sodium and calcium pulp are back towards their original site from where they originate. It should be maintained. And this cycle is repeated continuously in a sinusoidal pattern. I mean to say, one after again, one after next, one after next. I mean to say, the first the sodium ions will come on action, then calcium ion. Likewise, they are continuously acting on the muscle fibers, and they are making the fibers to contract or relax. So basically this calcium ion, this diffuses through the cytoplasm in 200 to 300 milliseconds, and that is the latent period between the initial excitation and beginning of contraction. To say whenever there is excitation or contraction, there should be a diffusion of calcium ions in range of 200 to 300 milliseconds. So it will initiate the excitation or the contraction of that specific muscle. And being this calcium is responsible for delaying the sequence because of slow process. As this calcium can consume 25% of oxygen in compared to skeletal muscle, because we are dealing with the smooth muscle. So this comparison is giving towards the skeletal muscle, that is only 25% of oxygen. So when there is action potential depolarizes to the threshold, and various transmitters which are released and diffuses the smooth cell membranes where stimulation occurs. When to say these neurotransmitters are basically this acetylcholine. So whenever this acetylcholine is released, the action potential will depolarize towards their threshold maximum level. And the innervation is usually conducted by the autonomic nervous system. I mean to say the parasympathetic and sympathetic nervous system. So in the skin, what happens? The pilomotor fibers, sweat glands, and cutaneous vessels, they receive only sympathetic innervation. While the innervation regarding the smooth muscle will conducted with the automatic nervous system having both the sympathetic and parasympathetic nervous system. But especially the skin, the sweat glands, the cutaneous vessels, they are innervated only by the sympathetic innervation. This acetylcholine that is released from the parasympathetic nerve fibers and norepinephrine from the sympathetic fibers. Okay. मतलब acetylcholine will enhance the intestinal peristalsis, whereas norepinephrine, it will inhibit the peristalsis. So say two actions is going to happen in the intestine, and both the function will innervated by autonomic nervous system, but from the different divisions. So here you can see the peristalsis will initiated by norepinephrine, that is the sympathetic, and antiperistalsis movement will be recovered by parasympathetic nerve fibers. So this is the things by which this smooth muscle will make contract or relaxed. So regarding the cardiac muscle, it will require definitely, generally what happens, the muscles have to either contract or relax. So for that, they required some nerve stimulation. But for cardiac muscle, they don't have requirement of any nerve stimulation because they have their SA node, that is also known as a pacemaker cells. And the cardiac muscle has also an intrinsic or inherent ability to generate their action potential rhythmically. Rhythmically means to say in a continuous nature. And basically this is done by the SA node. Exactly what happens? This SA node will depolarize faster than any other part of the heart muscle. And as it is, they generate the action potential. Mean to say they make the depolarization of the cells and get innervated and send various stimulation of heart via through this SA node. All the organs or all the parts of the heart will get innervated and it will contract or relaxed rhythmically. So regarding the function, it is limited because the regulation of heart rate is normally done by the pacemaker cells. So whenever there is requirement of contraction or relaxation, it is completely defined or designed by the pacemaker, that is a SA node. So basically this cardiac muscle action potential is 150 millisecond in the atria and almost double to it in the ventricles. So this contraction time, it lasts as long as the action potential does. Mean to say up to the limit when the calcium ions is come out from their original site or from their restoring site, it will depolarize. After that, it will repolarize. So this extended period, this will provide time for pumping the blood out of the ventricles. To say whenever there is a contraction in heart, the blood will pumping out of the ventricles and filling them again before the next beat. That will complete one cardiac output. There is a term hypertrophy that will tends to increase in cell size. And it is especially occurs in cardiac muscle when the heart has excessive work to do. If an animal is performing some exercise or heavy work, then the hypertrophy word will be prevailing because it covers that the size, the cell size of the muscle will definitely increased. Increased in the sense of quality, not the quantity. In man, human condition, sometimes it's also known as athlete's heart. One another topic, that is the brisket disease. So this brisket disease is also known as high mountain disease of cattle. Involves enlargement of the heart as well as edema of the brisket and involvement of the lungs. And to say this is Viscous disease, this is a can say normal condition in the animal, which comes after the adaptation of the animal in a certain surrounding, or especially in the high mountain. Because what happens in this condition, if an animal adapt to it, they have large number of hemoglobin size, and they have the uh, working efficiency. They have just made their enlarged, or they have the capacity. They tend their increase their capacity to make the inhalation and exhalation, as well as the enlargement of the heart, so that the more amount of blood can be conserved, and it will passes towards the all compartment of the heart. So it will helpful in view of continuous and more oxygenated blood towards the whole body. So basically, this is the figure which describes the intercalated disc and some myocytes present in the cardiac muscle. So you can have the picture that this they are having some interstitial space, that is also known as the intercalated disc, through which the SA node can efficiently work. This is the microscopic anatomy of cardiac muscle. So in picture A, you can see the nucleus, intercalated discs, cardiac muscle cell. In the next picture, the gap junctions are there. And the last picture, that is the fascia adherens. And to say, these are the sites by which the heart can produce their impulse and generate or to transfer it within all parts of heart rhythmically. This is the cross-sectional part of the cardiac muscle. You can have a very clear vision regarding this intercalated disc, the nucleus, mitochondria. Another view to reveal the T-tubules or sarcoplasmic reticulum, mitochondria, sarcolemma. Since these things are present in all types of muscle, but they are mostly prominent in the skeletal muscle. And it is present in also cardiac muscle, but it is not so much prominent. So regarding the skeletal muscle, contraction and relaxation. So this is the basic theme, because when an action potential generates along the muscle fiber, that is the sarcolemma, these fibers, they begin to contract after an initial latent period of about 3 milliseconds. That is a very low time. Is action potential generation of this action potential occurs within a very short time, that is 3 milli per second. So this action potential, that is initiated by the firing of a motor neuron. Mean to say, after getting the activated of T tubules through the actin and myosin filaments, and these actin and myosin filaments will get in action when this sodium and calcium ions, especially the calcium ions, will comes in action, then it will tend to move or contract or relax these T tubules, and further it will act for the generation of action potential, and that will tends to make a muscle for either to contract or to relax. So after these T tubules, when it gets in action, it will again generate its impulse to the longitudinal tubules, and after this, the circuit will be complete. So to complete the electrical circuit, this current transfers the wells and transfers the walls of the longitudinal tubules into the sarcoplasm and back outward through the whole cell membrane. So when getting the connection with these T tubules to the longitudinal tubules, the local circuit of ionic current flow will completed, and throughout the entire muscle fiber, the action potential will generate. So when this electrical potential applied to a T tubule at the surface of muscle fiber, it will cause contraction of the half of sarcomere supplied by the tubule, and the rest part will be transcribed by the longitudinal tubules. So one theory is also there, which should we keep in our mind, that is the all or none law. Perhaps previously in previous class I have also talked about this. I mean to say, if a muscle fiber will contract, then the whole muscle will contract. Or if a single muscle fiber will relax, then the whole muscle fiber will relax. I mean to say, if any contraction or relaxation is going to happen within a muscle fiber, then it will tend to make its action towards the whole muscle. So again, this picture, I think we have discussed it in previous class. So after getting this, the spread of electrical current inward through T tubules when an action potential passes has direct electrical effect on the interior of the muscle fiber to cause attraction between the actin and myosin filaments. So regarding the myosin and actin filaments, each thick filament in a sarcomere is a bundle of about 200 myosin molecules in two parts. That is light meromyosin and heavy meromyosin. So this LMM, light meromyosin, it lies parallel to other light meromyosin and make up the length of the whole thick filament. To say combining this light meromyosin, they will combine and form a whole thick filament. Whereas this heavy meromyosin, they will project outward like an arm from the end of the light meromyosin filaments. Meant to say, these heavy meromyosin, they comes in action like they are bounding this all the light meromyosin and acts as a single unit. So this arm projecting, it is flexible like a hinge where it joins the light meromyosin and also to the head of the other light meromyosin to head meromyosin. So this head attached to the free end of the arm and the complete hinged heavy meromyosin particles, it will called a cross bridge that protrude from all around the thick filaments. To say this cross bridges, they are performing through the hinged heavy meromyosin particles. Meant to say, when a series of light meromyosin are there and they are covered or be capped with the heavy meromyosin, they will collectively form a cross bridge which comes in action and protrude from all the thick filaments. So they away from the center in both directions. So there are no cross bridges in the center of the thick filament because it constitutes for the side by side light meromyosin for the attachment. So each thin filament is made up of three components, that is actin, tropomyosin and troponin. And the base unit is the F-actin molecule, which consists two long strands around each other in spiral. So each strand that is made up of hundreds of G-actin molecules. So here the G-actin molecules is very much essentially required for performing the action of this skeletal muscle. So lying in the groups of two F-actin spiral chains that are tropomyosin molecules, they will attach together to form a large strand. And the third protein, that is troponin, it will attach to each other tropomyosin molecule, and together are called troponin tropomyosin complex. So whenever there is contraction or relaxation will occur in regard to or in respect with these actin and myosin filaments, this troponin tropomyosin complex is very much or essentially required. It will lead to make the whole muscle in contact position or in relaxed position. So the presence of calcium ions, this causes the myosin molecules to develop an ATPase capability. I mean to say they will liberate some energy and make it the work done. Either it is going towards the retraction or contraction or relaxation. So there is one more theory, that is a ratchet theory, that will reveals the theory for muscle contraction. So basically what happens in resting state, the negative charges of the ATP bound with the cross bridges from different myosin filaments and the negative charge of the actin filaments. These causes the two to remain in the uncombined state with no attractive forces on both them. So on the appearance of calcium ions, the following amounts will occur. I mean to say the calcium ions bind with negative reactive sites of the ATP on the myosin cross bridge, and at the same time with the negative reactive sites on the actin filaments, which pulls these filaments together so that the muscle contraction could happen. And at the same time, the cross bridge, they are having the strong electronegativity in the resting state because of the presence of ATP. And when once this ATP will come out with the calcium ions, then again it will get relaxed. So this myosin filaments, they shank the filament negatively charged. Again, these calcium ions, they bind with the ATP on the cross bridge, and the negativity of the cross bridge become neutralized, as I say, and the contraction will change into the relaxed motion. So this bridge, they bend inward toward the axis of the myosin filament and pulls the axis of the myosin filament to shorten the muscle. So when these cross bridges fold in against the shank of the myosin filament, it will cause the splitting of ATP to ADP. And this breakup, the calcium ion will link between the myosin and actin cross bridge. And in the meantime, this actin filament will pulled along the axis of the myosin filament so as to contract the whole muscle. So subsequent similar reactions occur at the other cross bridges, and the actin filament is pulled to another state. So by doing these things, the energy from other sources like creatine phosphate, that will reconstitute the ADP to ATP. Again, it will be rebagged. And the cross bridges that has folded inward now bend outward again and bind with other calcium ions to pull the actin filament to next step. So this is about the ratchet theory. And regarding this relaxation, when muscle contraction will continue as long as there is an excess of calcium ions that will present in the sarcoplasm. But when the effect of the current spread at the triads end, this calcium ion will sequester back into the longitudinal tubules. After this sequestered of calcium ions, the sarcoplasmic reticulum will use the ATP energy to pump the calcium from the sarcoplasmic fluid back into the tubules and ready for the next deploration. Means the reploration action will occur after the back inward of calcium ions towards their T tubules position. So a small amount of calcium ion may be left out in the sarcoplasm, which acts as tone to create or to initiate the next depolarization. That is a very important thing. So it will also act for the compilation of this troponin tropomyosin complex. So during relaxation, the thin and thick filaments were dissociated, allowing the elasticity of the muscle to return into its resting length. And this will cause to pull the Z line and thin filaments back to their original positions. So this is the summary of excitation and contraction, whatever we have read this time, whatever we have discussed. This will compile the whole summary. The next topic should be effect of temperature. I mean to say, the climatic condition reveals regarding the contraction and relaxation of smooth muscles. So whenever there is increased temperature, the contraction will act somewhat fastly. But whenever there is a cold climate, the contraction will occur somewhat slow in comparison to the hot climate. So regarding this thing, whenever environment temperature is much below the normal body temperature, heat production by muscles is a definite advantage. That is, we also know it by shivering. And when air temperature is extremely low, these muscles may undergo spasmodic contractions called shivering to produce enough heat to maintain normal body temperature. So some points are regarding the stimulus for muscle contraction. So I think it is better to compile this topic in the